باشین. من واقعیتش برای جلسه امروز مطالب رو آماده کردم، ولی یه تعدادی فیلم هم از سایت‌های مختلف گرفتم. اگر موافق باشین ما اول اون فیلم ها رو ببینیم، بعد ادامه درس رو در خدمتتون هستم. این هفته سعی کردم هم برای بعد هر از چندگاهی البته فیلم رو متوقف میکنم، برمیگردم تو صفحه اصلی، چون من زمانی که فیلم رو بذارم پخش بشه نمیتونم پیام های شما رو ببینم. حالا مگر اینکه ببینم الان اگر صفحه رو کوچیکش بکنم بتونم همزمان پیام های شما رو هم ببینم. بسیار عالی میشه اگه به من یه چند لحظه فقط اجازه بدین. شما رو ببینم که اگر جایی وسط کار فیلم دچار مشکل شد بتونم متوقفش کنم. خب من صفحه‌مو شیر میکنم با شما. خب الان باید صفحه منو ببینید، درسته؟ الان باید شما صفحه منو ببینید. خیلی متشکرم، مرسی.
Hi, it's Dr. LeBlanc. This is our last video screen capture lecture in Unit 1, the molecules of heredity. In this unit, we're going to focus on the organization and packaging of DNA in eukaryotes. Our learning goals for this lecture are to understand the organization of functional sites on eukaryotic chromosomes, how eukaryotic chromatin is packaged, and how chromatin is organized in the nucleus. Eukaryotic genomes consist of a nuclear genome, which is packaged into chromatin and chromosomes. It also consists of mitochondrial and chloroplast genomes, which we're not going to discuss in this video lecture. A eukaryotic chromosome is one long strand of DNA from tip to tip. This is one long piece of DNA, unbroken strand of DNA from tip to tip of a eukaryotic chromosome. They can contain many origins of replication, approximately 100,000 base pairs apart. They have at the middle of the chromosome a centromere to which is attached a kinetic core, which is a complex of proteins that aid in making sure the two sister chromatids go to opposite poles during mitosis. They have telomeric regions on the ends of the chromosomes, which prevent shortening during DNA replication. And all of this DNA is wrapped around structures called nucleosomes. Nucleosomes aid in eukaryotic chromatin compaction. Nucleosomes are composed of an octamer, that means 8 histone proteins. And here is our basic structure. This is a nucleosome. These are the proteins, these orange blobs. There are 2 each of H2A, H2B, H3, and H4. It's a dimer of 2 each of these proteins. DNA gets wrapped around this ball of proteins, and then there's a linker region, and then another ball of proteins. Wrapping the DNA around nucleosomes creates a fiber that is 10 to 11 nanometers in diameter. I often refer to it as a 10 nanometer fiber rather than an 11 nanometer fiber. So a nucleosome consists of the 8 histone proteins and approximately 146 or 147 base pairs of DNA. The linker region in between the 2 nucleosomes can vary from 20 to 100 base pairs, depending upon the species and the cell type and the region of the chromosome that is being either transcribed or not transcribed. Here's a ribbon diagram of a nucleosome here. There are 2 molecules, each of H2A, H2B, and H3 and H4. And the DNA is wrapped around this. And you can see the relative structure or the relative size of the DNA versus the proteins. DNA is a very large molecule, and the proteins in the middle are a bit smaller. Also sticking out from the histone proteins, you'll notice these long tails. We'll get back to the function of these in future video lectures, but for now, know that these proteins' tails that stick out from the nucleosomes can be modified by acetylation, by methylation, by phosphorylation, and that those modifications will affect gene regulation. How is the structure of the nucleosome determined? Roger Kornberg, a very prominent DNA and chromatin biologist, proposed the model for nucleosome structure in 1974 based on his biochemical, X-ray diffraction studies, and electron microscopy images. Marcus Knowles' experiment, however, gave us a very visual and easily visually interpretable result to understand how DNA is wrapped around the nucleosomes. So his experiment began with nuclei, not extracted DNA, but actual nuclei. And you can remove nuclei from cells and work with them individually in test tubes. So when you have a nucleus, you have DNA that is associated with proteins. These orange balls each represent a single nucleosome, and the blue line is DNA. Treated these nuclei with 3 different concentrations of DNAs. Now, his idea was that the nucleosomes are going to protect the DNA, but the regions in between the nucleosomes are going to be digested by DNAs. At a high concentration of DNAs, every single linker region of this chromatin would be cut by DNAs. But at low concentrations, very low concentrations of DNA, only some of these regions would be cut. That would leave different lengths of DNA at the end of the digestion. Okay. After he treated the nuclei with the 3 different concentrations of DNAs, he treated the nuclei with detergent and phenol to extract the DNA, and he ran the DNA out on an agarose gel. Agarose gels are a matrix. If you apply an electric field across the agarose gel, the DNA will migrate towards the positive pole because DNA is negatively charged. The smaller fragments will migrate quicker and the larger fragments will migrate slower. So you can separate DNA based on its size by this technique. After the DNA has been run on the gel, you stain it with ethidiabromide, which intercalates into the double strands of DNA. It fluoresces under UV light, and you can take a picture. So this is how we interpret the data that Marcus Noll got at the end of his experiment. This is a length of chromatin. These are the nucleosomes, and these are the nucleosomal regions here, the linker regions. So when the DNA is cut by DNase, you can end up with a length of DNA that is one nucleosome's worth of DNA. At high concentrations of DNase, all the chromosomal DNA is digested into fragments that are 200 base pairs long. This is what the result he got. And this is how he interpreted the data, was that every single linker region was cut by the DNase, and that the length of DNA that was wrapped around a single nucleosome was 200 base pairs. At low concentrations, however, not all the linker DNA was cut. You may have cut here, and then here, but nowhere else. So you ended up with fragments of DNA that were 1 nucleosome long, or 2 nucleosomes long, 400 base pairs, or 3 nucleosomes long, or 4, 5, 6, 7, 8, etc. So in this way, Marcus Noll was able to determine that the DNA in the nucleus wrapped in chromatin was approximately 200 base pairs long around each nucleosome. In addition to the core nucleosomes, nucleosomal proteins in an octamer, a histone octamer, there are additional proteins that bind to DNA in the nucleus. There is another histone called H1 that binds to the DNA just next to the chromatin, next to the nucleosome, excuse me. There are also other non-histone proteins known as, there are DNA binding proteins known as non-histone proteins that play a role in organization and compaction of the chromatin. And this is a very large group of heterogeneous proteins. Lots of different kinds of proteins are called non-histone proteins. Without H1, the chromatin is a 10 nanometer fiber. With the addition of histone H1, you create a 30 nanometer fiber. And the 30 nanometer fiber is a coil of the 10 nanometer fiber chromatin. So there are two different models, and they probably both exist in the cell. A solenoid model, which is very compact. You have the nucleosomes wound tightly around each other, and a zigzag model, which is a bit looser form of chromatin, but they are both 30 nanometer fibers. Okay. So now we have our chromatin. We've gone from a DNA strand to a 10 nanometer fiber to a 30 nanometer fiber with the addition of H1. How do we get that 30 nanometer fiber to compact even further into chromosomes that exist in both the interphase nucleus and at the metaphase chromosomes that we see during mitosis? In a nucleus, there are many, many proteins. Nuclei are not an empty bag of fluid. They contain something called the nuclear matrix, which consists of lots of different types of proteins. Nuclear lamina, which is just under the inner membrane of the nucleus. There are scaffolding proteins, and there are matrix attachment proteins. This is an actual electron micrograph of a nucleus that has all its DNA removed. All the chromatin has been removed. And it's not empty. It has this matrix of proteins all around the inside. It looks a little bit like a sponge. This web of proteins still exists inside the nucleus, even if you take out all the DNA and the proteins that are bound to the DNA, like the nucleosomes. So just as prokaryotic DNA was organized into loops, eukaryotic DNA is also organized into loops. This green line represents a protein fiber of the matrix, of the scaffold of a chromosome, and the blue line, of course, is DNA. The loops can be quite variable in length, from 25 to 200 base pairs long. There are matrix attachment or scaffold attachment regions where the DNA is bound to the matrix or scaffold of the chromosome. And the Mars are anchored, the matrix attachment regions of the chromosome are attached to the nuclear matrix, creating these radial loops. So the DNA inside a nucleus of a eukaryote, although it may appear somewhat loosely organized, is actually quite organized. This is an electron micrograph showing a close-up of what the nuclear matrix looks like. Again, this has no DNA in it. This is just the nuclear matrix. Scaffolding of proteins that exist inside the nucleus. Chromosomes have different They have heterochromatic regions and and again at the telomeres. These regions of the chromosome are always heterochromatic. They are always tightly packaged, where the DNA is very tightly coiled around proteins. Chromatin exists in the other sections of the arms of the chromosomes, and that's where we find the genes that are being expressed. The Chromatin 30 nanometer fibers are anchored in radial loops in these regions of the chromosome. The heterochromatin is constituted by greater compaction of the radial loops. This is an interphase nucleus, stained so that each chromosome is a different color. So here we have chromosome 1 here, chromosome 2 in green, chromosome 3 in blue. It's interesting to notice that even in eukaryotic chromosomes, the DNA, eukaryotic nuclei, that the DNA from individual chromosomes is not intertwined with other chromosomes, but they remain in specific regions of the nucleus. This is a little bit like the nucleoids we saw in prokaryotes. Okay, so this is a very important figure because it shows the hierarchy of organization of eukaryotic chromatin. First, we have the DNA double helix, which is approximately 2 nanometers in diameter. Adding the histone core, notice it's a very complex multi-subunit protein that compacts or organizes the radial loops. Cohesin is found in between the two sister chromatids. Initially, when the metaphase chromosome is not as compact, and even further along during metaphase, where the chromosomes are even more compact at the beginning of prophase. Finally, in the middle of prophase, the cohesin molecules are dissociated from the sister chromatids, except for right at the center here, at the centromere in the kinetic core, and that's the last of the cohesin to during anaphase, when the two sister chromatids split apart and go to opposite poles into their new daughter cells. So in summary, eukaryotic DNA is packaged into chromatin. Nucleosomes are the fundamental unit. Nucleosomes are coiled into 30 nanometer fibers. Nucleosome organization was demonstrated by the digestion of chromatin with varying concentrations of DNAs. That's a very important experiment. You should make sure you understand that well. Radial loops attached to the DNA scaffold. Chromosomes form domains in the nucleus and attach to the nuclear matrix. And multiple proteins organize the chromatin into metaphase.
یه فیلم دیگه هم هست، اگر اجازه بدین این رو هم براتون پخش کنم. البته چند تاست.
In order to fit within a cell, DNA is packaged. DNA strands are wrapped around proteins and ultimately coiled over 1,000 times. This structure is called as chromatin, and proteins are called histones. There are 5 different kinds of histone fission. Chromatin condenses into visible structures called chromosomes. This electron micrograph shows chromosomes lined up in the middle of the cell. The presence of histones in nucleus of cells was part of debate about which molecule, DNA or protein, is hereditary material. Of course, DNA turned out to have that distinction. However, the X-ray diffraction studies later showed histones play an important role in providing structure for DNA helix. In 1964, Morris Wilkins and Vittorio Lazzari noticed that chromatin has a repeating pattern with intervals of about 100 angstroms. This repeat is different from repeating pattern of DNA itself. Based on X-ray diffraction pattern and nuclease experiments, the 200 base pair repeat observed after digestion, which corresponds to 200 base pair of DNA wrapped around each histone core. The 100 angstrom measurement from X-ray diffraction is the width of histone core and DNA. Roger Kornberg confirmed this model through experiments. He also figured out the arrangement of histones in the core. He individually purified the histones and figured out that H2A and H2B stick together as 2H3 and H4. If the H2A, H2B complex is mixed with H3, H4 complex with naked DNA, X-ray pattern for chromatin is achieved. More analysis revealed that each histone core has 8 proteins, 2 copies each of H2A, H2B complex and H3, H4 complex. This histone core with wrapped DNA is called nucleosome. This is an electron micrograph of chromatin. This fiber is called 10 nanometer fiber. But where is H1 histone? It turns out that H1 histone is not part of histone core. Instead, it binds nucleosomes to give even more structure to chromatin. H1 histone sits just outside of each nucleosome and interacts with the H1 of the other nucleosome. At higher salt concentrations, 10 nanometer is further compacted into 30 nanometer fiber. The DNA structure is already twisted. By adding more twists to nucleosomes and solenoid structures, DNA is supercoiled. Even more organization is involved in maintaining condensed chromosomes. Non-histone protein makes scaffold proteins, which maintain shape of chromosomes even in absence of histones. Up to 6 feet of DNA is packaged to fit into nucleus of cell. The DNA is wrapped around histone cores to form nucleosome and 10 nanometer fiber. The 10 nanometer fiber is further coiled to form 30 nanometer fiber, where 6 nucleosomes make one turn. The 30 nanometer then looped into scaffold proteins when chromosome condenses. DNA packaging has an important role in gene expression.
من عذرخواهی میکنم یه لحظه از اینترنت قطع شدم. الان داره صفحه رو شیر میکنه که اون تیکه آخرش رو که صفحه رو اومد رو دوباره بتونم خدمت شما نشون بدم. وقف کرد شیر رو. درست شد. فکر می‌کنم از این قسمت‌هاش بود که این صفحه اومد منم نتونستم بهش دست بزنم.
complex and H3, H4 complex. This histone core with wrapped DNA is called nucleosome. This is an electron micrograph of chromatin. This fiber is called 10 nanometer fiber. But where is H1 histone? It turns out that H1 histone is not part of histone core. Instead, it binds nucleosomes to give even more structure to chromatin. H1 histone sits just outside of each nucleosome and interacts with the H1 of the other nucleosome. At higher salt concentrations, 10 nanometer is further compacted into 30 nanometer fiber. The DNA structure is already twisted. By adding more twists to nucleosomes and solenoid structures, DNA is supercoiled. Even more organization is involved in maintaining condensed chromosomes. Non-histone protein makes scaffold proteins, which maintain shape of chromosomes even in absence of histones. Up to 6 feet of DNA is packaged to fit into nucleus of cell. The DNA is wrapped around histone cores to form nucleosome and 10 nanometer fiber. The 10 nanometer fiber is further coiled to form 30 nanometer fiber, where 6 nucleosomes make one turn. The 30 nanometer then looped into scaffold proteins when chromosome condenses. DNA packaging has an important role in gene expression.
بعد
Did you know that the length of a DNA or deoxyribonucleic acid double helix in a typical mammalian cell is about 2.2 meters? This length is obtained by multiplying the total number of base pairs present in the DNA double helix with the distance between two consecutive base pairs. Now, the total number of base pairs present in a DNA double helix in a typical mammalian cell is 6.6 multiplied by 10 raised to the power 9, and the distance between two consecutive base pairs is 0.34 nanometers, or 0.34 multiplied by 10 raised to the power minus 9 meters. By multiplying these two factors, we get the length of the DNA double helix as 2.2 meters. Have you wondered how such a long polymer is packaged within a typical nucleus with a dimension of about 10 raised to the power minus 6 meters? To understand this, let us learn about the packaging of a DNA double helix, which is different for both prokaryotes and eukaryotes. In prokaryotes such as E. coli, the nucleus is not very well defined. However, the negatively charged DNA is not scattered throughout the cell, but is arranged in large loops, is held together by a few positively charged proteins in a region known as the nucleoid. On the other hand, in eukaryotes, the manner in which a DNA double helix is packaged within the cell is more complex. Eukaryotes contain a single molecule of negatively charged DNA packaged around a spool of positively charged basic proteins known as histones. The charge in a protein is decided by the abundance of amino acid residues with charged side chains. Now in this case, the proteins are positively charged as the histones are rich in basic amino acid residues like lysins and arginines, which carry positive charges in their side chains. Furthermore, the histones are arranged to form a unit of 8 molecules known as the histone octamer. Now, around the positively charged octamer, negatively charged DNA molecule is wrapped to form a structure known as the nucleosome, which is held in place by the H1 histone. A typical nucleosome has around 200 base pairs of DNA helix, and the nucleosome nucleosomes present in chromatin can be seen as a beads-on-string structure when observed under an electron microscope. It is the nucleosomes that make up the repeating unit in chromatin, the thread-like bodies present in nucleus. Now, the chromatin fibers are of two types, euchromatin and heterochromatin. In a typical nucleus, euchromatin fibers of 30 to 80 nanometers in diameter are loosely packed and stain light. On the other hand, heterochromatin fibers of about 300 nanometers in diameter are more densely packed and stain dark. Moreover, of the two types of chromatin fibers, euchromatin is transcriptionally active, while heterochromatin is inactive. These chromatin fibers coil further and condense to form short and thick bodies called chromosomes during the metaphase stage of cell division. In this way, a DNA double helix is packaged into a chromosome, which is further packaged within the nucleus.
اینها بچه‌ها یه تعدادی فیلم هست که به اون بحث قبلی ما که در مورد مدل‌های مختلف کروماتین هست به درک بهترش همشون کمک میکنه. اینم باز یکی دیگه هست.
Epigenetics refers to the heritable modifications in change is histone modification that may be caused by overexpression or aberrant recruitment of HDACs. Within the chromosome, DNA is packaged into chromatin. Chromatin consists of DNA, structural histone proteins, and non-histone proteins. Within chromatin, the repeating unit is the nucleosome. Nucleosomes are made up of 146 base pairs of two superhelical turns of DNA wrapped around a core of 8 histones. The histones are responsible for maintaining the chromatin's shape and structure. Epigenetic modifications, such as histone acetylation, occur at the amino-terminal tails of the histones that protrude from the nucleosomes. Acetylation of histones is generally acknowledged to play a key role in the regulation of gene expression. Histone acetylation is controlled by the balance in the activity of two enzymes, histone acetyltransferase, or HAT, and histone deacetylase, or HDAC. For a gene to be transcribed, it must become physically accessible to transcriptional machinery. Acetylation of histones by HAT causes uncoiling of DNA and an open chromatin structure. This causes genes to become accessible to transcription factors, allowing gene expression to occur and proteins to be made. Conversely, deacetylation of histones by HDAC results in tight coiling of the DNA and a closed chromatin structure. In some cancer cells, there is an overexpression of HDACs, an aberrant recruitment of HDACs, or an underexpression of HATs, resulting in hypoacetylation of histones, and therefore a condensed or closed chromatin structure.
بعد نمی‌کرد دیگه. الان با توجه به این فیلمایی که دیدین و توضیحاتی هم که جلسه گذشته خدمتتون ارائه دادم، خیلی خوب دیگه میتونید این رو درک کنید، مخصوصا دانشجویانی که رشته پایه‌شون زیست شناسی نبوده. قرار گرفتن اکتامرهای هیستونی دور تا در داخل دی ان ای و همچنین حضور هیستون‌های یک، مدل سولنویدی و همچنین مدل زیگزاگی که توی اون دوتا فیلم قبل‌تری خیلی خوب اینا رو میتونست نشون بده که باز توی این فیلم هم باز داره همین رو نمایش میده. مدل سولنویدی رو شما دارید میبینید و سطوح بالاتر کروماتین که حالا به تدریج که فیلم داره جلوتر میره اینا رو نمایش میده. اسکافولد هایی که در داخل هسته هستن و اینها روشون قرار میگیرن به صورت متصل به اسکافولد های داخل هسته. به وجود اومدن سطوح بالاتر کروماتین 300 نانومتری 700 نانومتری این در سطح 700 نانومتری هست. خواهش میکنم خانم بیاتی، متشکرم، نظر لطف شما. بازم البته من یه بار دیگه چک بکنم فیلمایی که نشون دادم رو. یه فیلم دیگه هم بود بله این فیلم خیلی جالبیه راجع به مودیفیکیشن های هیستونی هست. البته ما راجبش صحبت نکردیم ولی حالا بد نیست این رو هم ببینیم.
As well as modification to the histone tails that emanate from the core of the nucleosome. The tails of the core histones labeled here can be altered with distinct chemical modifications. Examples of these modifications include methylation for histone H3, acetylation for histone H4, and phosphorylation for histone H2b. New chromatin is often characterized by a more open and accessible state of the DNA, one in which transcription factors have access to their cognate binding sites, and can therefore recruit enzymes like histone acetyltransferases that acetylate histone tails, and activate genes by recruiting components of vessel transcriptional machinery, including RNA polymerase. Heterochromatin, in contrast, is thought to be characterized by a more tight bundling of nucleosomes, which impedes transcription factors from gaining access to regulatory sites on the DNA. Methylation of cytosine bases in regions called CPG islands is a hallmark of transcriptionally repressed heterochromatin. These methylated cytosines, in turn, recruit proteins like MECP2, methyl CPG binding protein 2, and HP1, heterochromatin protein 1. These proteins are thought to maintain a repressive state of chromatin by inducing histone deacetylation by HDACs, as well as histone tail methylation by histone methyltransferase enzymes.
این فیلم که دیدین اگر دقت میکردین بحث هتروکروماتین و یوکاروماتین رو مطرح میکرد، بعد که شما البته به این مفصلی نمیخونید. این هم باز یه فیلم دیگه هستش.
Welcome to video 5, DNA packaging and chromosomes. Now, if you're watching this during Valentine's Day,
اینو که ندیدین بچه‌ها، درسته؟
great. So, I want to start off by talking about this drawing really quick. This is really sad, actually. This drawing, whoever made it, obviously did not know his biology or her biology because DNA does not look like this. These are actually supposed to be hydrogen bonds, which are invisible. They're bonds, but here they're little beads, like candy. You're thinking of Valentine's.
درسته خانم بیاتی، من اینو نذاشتم. بله، مرسی.
Something about Valentine's Day portion selection. Here we go. Okay, so we're going to look at chromatin, chromosomes, and chromatids today. They all sound like chroma words, but I'm going to say that it's really important to know the distinction between the three. In fact, you'll find that even though they're very similar, they're going to be referring to different phases of mitosis. So we do need to be familiar with them, especially as we continue to study cancer. and birth defects and other things. So I want to start by looking at this beautiful cell here. If you look here, the cell, you can notice the nucleus with all its little DNA inside. Yeah, look at that beautiful worm. And these DNA pieces are going to be the chromosomes of the cell. Now, chromosome and DNA are not actually synonymous. Right now, you're like, whoa, 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 you taught us that DNA is in the nucleus. But I'm going to tell you right now that there's a slight difference between chromosomes and DNA. And it has to do with the fact that proteins are involved. I do want to bring a little bit of attention here to this number 50 micrometers. Micrometers is actually the same as saying 0.000001 meters, or 10 to the negative 6 meters. So this is a very, very small number. The nucleus is about the size of 50 micrometers. And I want to keep that for reference because if you look at this next figure right here, this is going to show the levels of DNA packaging and how tightly DNA is condensing. So walking through this, if you look at this first picture on the top right here, this is going to be our plain double-stranded, double helix DNA. Right? This is the deoxyribonucleic acid we've all grown up to love. However, lies, the chromosomes in your body and in your nucleus are not actually just pure DNA. They're associated with proteins. So if you see these yellow things right here, these are actually histone molecules. These are histone proteins. So these yellow proteins are actually going to be anchor points for the DNA to wrap around. So this little red section here, this is raw DNA right here, this red section here, and this is also raw DNA. But what they're going to do is this raw DNA is going to wrap around histone proteins. So it makes a strange coiling motion like this. Okay. So again, all these pieces right here, maybe I should even color it with a different color. These are going to be histone proteins. Okay. And the DNA, the double-strand DNA, is going to be wrapping around them. And you're like, what's the point of that? Well, it allows us to condense it even further. So you can notice that these right here are going to be condensed even further here. Just a quick note that you might want to find helpful, is that when you have 8 histones in a row, 1, 2, 3, 4, 5, 6, 7, 8. When you have histones in a row, that is actually called a nucleosome. You're like, nucleosome? Well, that almost sounds like chromosome. Almost there. So if we had 8 of these, we only show 3 here. If we had 8 of these, that would be a nucleosome. Just another term for you to memorize. So right here, I'm actually going to undo a little bit. Okay. So from here, we can actually package it further. So we've gone from these partial nucleosomes to these very condensed forms. You can see right here. And this will condense even further here to this new coil right here. And this coil right here represented can condense even further into this big, fat, twisty thingy. And that thingy is eventually going to make up this small section of the chromosome. So basically what we've done is we've packaged DNA so condensed-like so that eventually it becomes a 10,000 times smaller size than it would be if we just stretched out on its normal length. So let's just recap really fast. Okay, I'm going to take away all the pictures. Okay, so just a quick recap. DNA by itself, right, just a double-stranded version, is way too big and way too long to just keep on its own. We've got to condense it to the much, much smaller form of the chromosome. So already right now I've introduced the term chromosome. That's going to be the highly condensed version of DNA. To help us do this, we need to use proteins. These proteins right here. And so you'll notice that the term chromatin appears here. Chromatin is basically the term we use to show how DNA is associating with the proteins. So let's actually do the definitions right here. Chromatin is simply DNA interacting with proteins, or DNA wrapped around proteins. And if you remember, it's basically this idea, where you have the DNA wrapped around a histone protein. So when DNA and histone proteins interact with each other, basically the DNA wraps histone, that is called chromatin. So a lot of people just kind of take the definition as DNA plus protein is chromatin. Chromosomes, on the other hand, are highly condensed chromatin. So this condensed like crazy into that super thick, small, stubby shape, that's going to be the chromosome. Okay? And the reason why this is important is chromatin will become a chromosome when it's time for mitosis. Right? When it's time for mitosis, we're not interested in gene expression. We're not interested in having all this DNA free. We just want it to be packaged in as small of a space as possible so that when we separate, we can keep everything nice and neat together. Finally, chromatids are kind of another beast completely, and this is going to be important for when we eventually enter S phase. So you should have remembered in the cell cycle that we covered already that there are four main phases. We've got the G1, S, G2, and the mitotic phase. And interphase kind of takes over. Phase kind of takes over these three G1, S, and G2 phases. Now, before we even get into G2, in S phase we have DNA synthesis. So for every strand of DNA that we have to make a copy of that strand, right? So that we can have twice the amount of DNA, so that eventually when we have two cells, each one will have a complete set of DNA. So let's pretend I have an organism that just has 3 chromosomes, right? So this is my cell with 3 chromosomes. Humans have 46, but I don't want to draw 46 chromosomes. So this is going to be an organism that has 3 chromosomes. When we eventually divide, we need to make sure that we have a copy of each of the chromosomes, right? And so that way, we can actually end up with 2 cells that each have their 3 original chromosomes. And I'm using different colors just to show how they might have split. And so chromatin actually refers uniquely to the point where we have 2 different chromosomes, right? And instead of calling it original copy chromosome and copy version of chromosome, we call each one a chromatid. Chromatid. It's really chromatid, actually, chromatid. So this is going to be one sister chromatid, and this is going to be the other sister chromatid. So we will cover more why chromatids are important when we talk about meiosis, actually, and when we go over the exact steps of mitosis. With that said, I'm going to just leave you with one last word, and that's the difference between somatic cells and gametes. Somatic cells refer to just your general body cells, right? So most of your cells in your body are going to be somatic cells, and soma actually stands for body in original language, and gametes actually stand for sex cells. And the reason why I'm bringing this up is we are going to be focusing on the division of body cells when we talk about mitosis, and we will be focusing on the division into sex cells when we talk about meiosis, and that's just a sneak peek into the next few weeks. So with that said, that's the video. Have a good day. Have a
خب فکر میکنم بقیه هم خدمت شما نشون دادم همه فیلم ها رو. امیدوارم که این فیلم ها مفید واقع شده باشه. یه جورایی تکمیل کننده مباحثی بود که در جلسه گذشته راجع به کروماتین، بعد مدل های مختلفش صحبت کردم خدمتتون که حالا به زبان انگلیسی هم توضیح میداد. انشالله اگر سوالی موردی باشه حل شده باشه. خب من واقعیتش این جلسه رو خواستم یک آنتراکی باشه بین بقیه جلساتی که تا الان داشتیم. توی کلاس بیوشیمی هم جلسه ساعت بعد ما بیوشیمی سلولی داریم بعد اگر یه چند لحظه باز به من اجازه بدید که ببینم برای ساعت بعدی دانشجویی که باز با من کلاس دارن، باز یه چند تا فیلم هست که باز اونا رو هم تهیه کردم براتون پخش میکنم که یه جورایی تکمیل بحث آپوپتوز باشه. یه جورایی ما بتونیم یک وقتی ایجاد بکنیم که انقدر هول جلسات پیش نره، بعد یک گپی هم باشه این وسط که باعث ایجاد تنوع توی کلاس بشه. خب جلسه گذشته من اینجا مشخص کردم که تا انتهای اسلاید 74 خدمتتون توضیح داده بودم. که خیلی کوتاه هم یه درس کوچیکی ما بدیم، بعد جلسه رو به اتمام میرسونم. ما جلسه قبل من البته این اسلاید رو خدمتتون توضیح دادم. این یک مدلی بود برای عملکرد دومین انتهایی سی ترمینال تو زیرواحد آلفا که به نام همون CTD معروف هستش. بعد مشخص میکرد که چون ما اومدیم راجع به عملکردهای مختلف زیرواحدها در RNA پلیمراز در پروکاریوتا خدمتتون شروع کردم به توضیح دادم. بعد اومدیم از زیرواحد سیگما شروع کردیم. فیلم ها رو واقعیتش من هیچ وقت به دانشجوها نمیدم آقای هرموشی. فیلم ها در اختیارتون قرار نمیگیره. البته خیلی هم راحت شما میتونید خودتون خیلی بهتر از این فیلم ها رو بگیرید از اینترنت و یوتیوب. بعد هم واقعیتش تو از طریق ایمیل ارسال کردن خیلی مشکله، خیلی وقت گیره. برای همین حالا تو بیوشیمی سلولی هم میبینید واقعا نفسگیره چون خیلی زمان میبره. بعد خیلی هاشون حجیمن. از طریق ایمیل متاسفانه نمیتونم در اختیار دانشجوها قرار بدم چون ظرفیتش خیلی زیاده. برای همین کلا الان چندین ترمه که دیگه منصرف شدم از ارسال فیلم برای دانشجو، چون اینا همه تو یوتیوب هم هست، شما میتونید اینا رو بگیرید حتی خیلی بهتر و کاملتر. چون فیلمایی بود مثلا 1.5 ساعت مثلا از سمینارها بود، خیلی دوست داشتم اونا رو بگیرم براتون نمایش بدم، ولی واقعیتش حجمش به قدری زیاد بود که دیگه برام امکان پذیر نبود که بخوام اینا رو بیام دانلود کنم. بعد تو فکرم هم بود که براتون ارسال کنم، ولی بعداً کلا منصرف شدم از این کار چون خیلی وقت میبره. به نت پرسرعت هم داره، بعد وسط کار قطع و وصل میشه. در حد فیلم‌های 1-2 دقیقه‌ای رو میشه ارسال کرد برای دانشجو، ولی اینا رو هم همونجور که دیدید بیشتر از 5 دقیقه است. برای همین واقعاً شرمنده نمیتونم اینا رو ارسال کنم براتون. بعد ما راجع به دومین های سی ترمینال و این ترمینال زیرواحد آلفا خدمتون توضیح دادم که حالا به نام آلفا CTD و آلفا NTD میگن که اینها هر کدومشون به صورت مجزا تا خوردگی توشون ایجاد میشه و 2 تا دومین مستقل ایجاد میکنن و یک رابط انعطاف پذیر هم اینا رو میاد به هم دیگه وصل میکنه. دومین هم تعریفش به این صورت هستش که بخش‌های از پروتئین هستن که میتونن به صورت مستقل دچار فولدینگ بشن که حالا اینجا چون اینا به صورت مستقل هستن ما اینا رو به عنوان دومین بهشون میگیم. RNA پلیمراز از طریق زیرواحد فاکتور سیگما خودش و بدون هیچ کمکی از ناحیه سی ترمینال زیرواحد آلفا میتونه به پروموتر مرکزی متصل بشه. اما اگر پروموتر دارای آپ المنت باشه اون وقت هستش که باید دومین سی ترمینال هم بهش کمک بکنه. بعد زیرواحد آلفا وارد عمل میشه و این یک ارتباط قوی رو بین زیرواحد سیگما و آپ المنت رو زیرواحد آلفا میتونه ایجاد بکنه و رونویسی رو امکان پذیر میکنه. و فکر میکنم یه مورد دیگه هم راجع به جداسازیشون فکر میکنم گفته بودم، نمیدونم، مطمئن نیستم. حالا بگذاریم میریم سراغ اسلاید بعدی یا طویل سازی هست، elongation، که بعد از زمانی که شروع رونویسی خاتمه پیدا کرد، حالا هسته آنزیمی شروع میکنه به طویل سازی RNA که اون وقت اینجا ما زیرواحدهای بتا و بتا پرین رو به طور خاص خواهیم داشت و راجع به جزئیاتشون تک تکشون انشالله صحبت خواهم کرد براتون. ببینید تا الان اون چیزی که خدمت شما توضیح دادم به خاطر اهمیت فاکتور سیگما تو تعیین اختصاصی بودن مرحله شروع رونویسی، خب خیلی از محققین روش تاکید کردن. ما هم از همین برنامه فاکتور سیگما شروع کردیم. ولی هسته پلیمراز میدونیم که داره در واقع ساخت RNA رو انجام میده. پس بنابراین هسته آنزیمی جزء بازیگران اصلی تو فرایند طویل سازی هستن. زیرواحدهای بتا و بتا پیرین توی تشکیل پیوندهای فسفودی استر دخالت بسیار زیادی دارند و این زیرواحدها در اتصال به دی ان ای هم نقش دارن. حالا مثلا زیرواحد آلفا میتونه کارهای دیگه هم انجام بده، مثلا مثل مونتاژ کردن هسته پلیمرازی. خب توضیحات نقش بتا و اینکه چگونه پیوند فسفودی استر رو میتونه برقرار کنه رو میذارم برای جلسه آینده انشالله توضیح بدم خدمتتون. یه حضور غیاب فقط خدمت شما انجام بدم، بعد کلاس رو به اتمام میرسونیم. خالد زوین حمید شویلی نبود؟ حضور ندارن الان تو کلاس؟ هستن ولی کنم صدای